Almost everyone's research, it seems like, involves high-throughput sequence data generation and analysis in some way.
We're giving them the skill sets and knowledge to take those bioinformatics dataset analysis skills back to their research and advance their own work and the field. We have 44 CRI Fellows, 5 faculty and 9 teaching assistants.
We're in this era of technology where we really have to make sense of it and think about the important biological questions that can be asked of these data.
The individuals in there are studying cancer, they're studying cancer immunology, cancer genomics, cancer biology, and the tools that we have increasingly available to do that are these high throughput tools like sequencing, mass spec, and so on that produce these giant data sets.
It's been a very collaborative process, both within CRI and outside CRI, in terms of how do we design a program that best fits the needs of CRI Fellows.
The trainees have come in with their own data sets, their own ideas, their own goals, and in the process we've been able to start from the foundation of building coding skills, building fundamentals in terms of how to run code and how to effectively apply it to a question.
So having the skills to do this analysis, especially if you're someone who works in the wet lab, is kind of critical, right? Analyzing your own data makes it very powerful for them.
Cancer immunology is a big field at this point. It's one of the advantages of training in these technologies. The second you realize what they're capable of, you can understand how they can be applied to all of these different questions and how they can support this very, very diverse field.
The sort of scale and complexity of data that biologists are now able to generate from their samples and for their experiments just keeps growing. So enabling people to take the data analysis into their own hands, I think is really empowering and really important to people starting a new lab that are going to be, training their own trainees and working with these increasingly sort of large and complex data sets.
So I'm very sure I'll be using sequencing as part of my projects in the future, and so this will allow me to have a bigger depth of understanding of that work.
It's not about having just one set of tools available, it's understanding how the field is adapting and growing and adapting to that.
There's no wet lab and there's no computational. They need to be together. They're intertwined now.